definitely the pleasure is mine. And first and foremost, thank you very much, like, for all the organizers here for the invitation and the opportunity to speak a little bit about Vigil Neuroscience, our current pipeline and our programs, and that's what I'm going to do over the next few minutes. Okay, or?
Dr. Meier, I think something that you're doing is hitting into the microphone. It's making a little.
Can you hear me better now?
We can hear you now. Yep.
Oh, okay, perfect. Yeah, sorry for that. I don't know what happened. It would be great if you could advance to the next slide.
All right, so let's advance the slide.
Yeah.
Thank you.
That's our forward-looking statements and some disclaimers, so we could just move right to the next. So maybe for the start, a few words on Vigil Neuroscience. Vigil Neuroscience was founded now, I think it's 2 years and 2 weeks ago. So it's a young company. It's based right in the heart of Cambridge, Massachusetts, right in Kendall Square. But even though Vigil is a young company, it was founded with a clear mission in mind, namely to to treat rare and common neurodegenerative diseases by restoring the vigilance of microglia, which are the brain's sentinel immune cells. And in this, Vigil is seeking to employ the tools which are currently available to us in modern neuroscience drug development to deliver therapies to improve the lives of our patients and their families. If you go to the next slide, please. With regard to our mission, we see it as our mission to discover and develop therapeutics that activate and restore the function of microglia by utilizing a precision-based development strategy. Our initial focus in this is on rare microgliopathies, and we have plans beyond this to also advance into broader indications in larger patient populations. Our first treatment candidates and development candidates target a key microglial receptor protein, namely the TREM2 or triggering receptor expressed on myeloid cells 2 receptor. And go to the next slide and speak a little bit more to our current portfolio. So concerning our current pipeline, we have here shown on the left-hand side our lead clinical development program for the treatment of ALSP with our monoclonal TREM2 antibody, VGL101. But furthermore, we are also developing, here shown on the right-hand side, a small molecule TREM2 agonist, which is currently in IND-enabling studies, and which, as an orally available therapeutic, offers, we think, great potential for larger indications, including larger neurodegenerative disorders like Alzheimer's disease, for example. And having these two modalities, we think, enables us to really pursue a robust pipeline in multiple rare and common neurodegenerative disorders. Move on to the next slide. So this just gives you a quick overview of our current development pipeline, here with our lead program, VGL101, on the top. With VGL101 specifically, we have made significant progress in implementing our strategy. We have an ongoing phase 1 study of VGL101 in healthy volunteers currently. This study is designed to specifically support subsequent patient trials in ALSP, but also other potential future indications for VGL101. And most importantly, with regard to the phase 1 study, so this puts us on track to initiate our first phase 2 trial in ALSP patients in the fourth quarter of this year. So this will be a very significant milestone as well for us as a company. And beyond this, as mentioned, Vigil is seeking as well to proceed in indications beyond ALSP. including rare indications such as CALD here or larger neurodegenerative disorders like Alzheimer's disease. Move on to the next slide, please. So maybe since we are a microglia-focused company, a few words on microglia. Microglia are the resident immune cells in the brain. And microglia has multiple functions. Some of them are shown here in the picture on the left-hand side. But specifically what microglia does is the recognition and elimination of foreign invaders, anything which doesn't belong there in the brain, and the repair of local damage to CNS tissue resulting from injury, for example. In addition, microglia are important for the overall neural circuit modulation and very relevant for maintaining like overall brain homeostasis. Conversely, what is known is that if microglia is dysfunctional, that this is associated with multiple rare and common neurodegenerative disorders. Could go to the next slide, please. So now, as I mentioned, we are focused on TREM2 as a therapeutic target. And back to microglia. So one of the key microglial receptor proteins, which is richly expressed on the surface of microglia, is the TREM2 receptor. And this is also the target of our lead asset of VGL101. And how this TREM2 receptor functions, maybe a little bit more on this, as you can see here on the left-hand side. So once activated, e.g. by like cell debris, foreign materials, the TREM2 receptor activates its associated transmembrane adapter protein, which is called DAP12. And this one, DAP12, then in turn subsequently activates recruitment of SYK or spleen tyrosine kinase here in the cytoplasm. So the receptor sits on the surface of the cell. We have the DAP12 protein basically crossing the cell membrane, and then we have the SYK recruitment in the cytoplasm. And this is important because SYK then causes the activation of multiple downstream signaling mediators, which then initiate a coordinated response to the problem. And this mechanism is essential for proper microglia function. And as shown here on the right-hand side, it also supports the transition of homeostatic, non-active, not overly active kind of microglia into the state of active neuroprotective disease-associated microglia, also called DAM. And here on the very right, loss of function of TREM2 is known to lead to dysfunctional microglia, and this then in the long term can have consequences like neurodegeneration. There is a disorder specifically related, Nasu-Hakola, which is another rare condition specifically related to TREM2 dysfunction. So TREM2 is a key receptor for like the proper functioning of microglia and homeostasis in the brain as a system. Move on to the next slide. Yeah, so for the remainder of this presentation, I want to provide a little bit more details around ALSP and our VGL101 TREM2 agonist lead program, specifically in what we are doing for ALSP. So go to the next slide, please. ALSP, or adult onset leukoencephalopathy with spheroids and pigmented glia, is a rare adult onset leukodystrophy. Mean age of onset is usually in a person's early 40s, but the range is pretty broad. It affects approximately around 10,000 individuals in the US. ALSP is a hereditary condition with an autosomal dominant pattern of inheritance, and it is known to be caused by a mutation in the CSF1R gene, or colony stimulating factor 1 receptor gene. And with regard to its clinical presentation, so the clinical picture of ALSP, which often makes the diagnosis, specifically in the beginning, difficult, can include various symptoms. What is very common, though, are cognitive impairment, often with a presentation similar to frontotemporal dementia. And in addition, movement disorder-like symptoms, seizures, like those can also often be observed in ALSP patients. And most importantly, to date, there is no approved specific treatment available for patients with ALSP. And if we go to the next slide, the question now is how an antibody like VGL101, which targets the TREM2 receptor, can benefit ALSP. So as mentioned on the previous slide, ALSP is caused by a mutation in the CSF1R gene. The CSF1R gene contains the information for another important receptor on microglia, namely the CSF1 receptor. And just like TREM2, CSF1R has essential functions as well important for microglial survival and homeostasis. A mutation now in the CSF1R gene leads to dysfunctional CSF1R receptors, which then over time is a cause for neurodegeneration and leads to the symptoms seen in ALSP. So now in order to understand like how TREM2 can potentially help ALSP patients, it is important to look at how the CSF1R receptor protein works, and as shown here on the right-hand side, a little bit towards the left. You can see that just like the TREM2 receptor, the CSF1R receptor also basically sits on the surface of microglia and the extracellular space. And exactly like the TREM2 receptor, also activates the DAP12 protein, which then in turn activates the spleen tyrosine kinase, or SYK, which means that both receptors ultimately share a common signaling pathway. And the hypothesis now is that by activating TREM2, which again activates the same kind of DAP2 and SYK cascade, the impaired microglial signaling activity in ALSP could be improved, and microglia function can be potentially rescued or restored. Go to the next slide. So since ALSP is an increasingly recognized disorder, but it is, as said, often specifically in the beginning, difficult to diagnose, and there's still a lot to learn. But Vigil has done is we have initiated a natural history study in ALSP in order to better understand the condition and to learn more about specifically the course of this disorder. And in this natural history study, ALSP patients with a documented mutation in the CSF1R gene who have clinical symptoms, or patients with an existing mutation in CSF1R and findings in their MRI alone, something which we kind of call prodromal at the moment, those patients can participate in the natural history study. The study is a global study. It is conducted in several countries, including the US, UK, Netherlands, and Germany. And if there should be patients in other countries, there are options for travel support to organize like a trip and travel to one of the existing centers. To the next slide. So the natural history study itself will last approximately 25 months and has planned study visits every 6 months. At these regular visits, clinical assessments will be performed along with MRI scans, lab tests, and a couple of other assessments which are included. As said, there are travel options for participants, potential participants who do not live close to one of the established trial sites. And also important to mention is that participating in the natural history study does not preclude participants from enrolling in the separate experimental interventional trial with VGL-101, which is planned to start in the fourth quarter of this year. So the next slide. In addition to the natural history study, there's also the option for participating in a registry, an ALSP registry. This is an app-based registry. It is currently available in the US, the UK, Netherlands, and in Germany. The purpose for this registry is to, again, learn more about ALSP, but in contrast to the natural history study, so everybody who has been diagnosed with the CSF1R mutation, symptomatic or asymptomatic carrier, can participate in the registry. Practically how it works is that, as said, it is an app available for Google and like for Android and iPhones. And through this app, there will be a survey about every 3 months, which is about like 30 minutes in length, just to kind of collect like general information around like how the individuals are doing and how ALSP or the knowledge of positive carrier status affects them and their family and friends. So with this, I think I'm at the end of my presentation. Yeah, I hope I could give you with this a little bit of an idea of what Vigil and what we are doing and our current clinical program. And I want to thank you for your attention and for listening. And I'm happy to answer questions if there should be any.